Hi, I'm Catherine Mishka, and it is November 7th, 2022. So in this unit, we started looking at energy metabolism pathways and hormone signaling of these pathways. So for this case study, I looked at case study number 3 and number 4. For case study number 3, insulin is a major hormone responsible for the uptake of blood glucose into organ, muscle, and tissue. In adipose and muscle tissue specifically, insulin stimulates specific glucose transporters in the cell membrane, which increases the amount of glucose taken in to be converted to energy. Insulin levels also work to direct the release of glucagon, which stimulates metabolism of alternate energy stores in the liver to produce glucose when the body is in a fasted state. When there's a lack of insulin presence in the bloodstream, glucose uptake by adipose and muscle tissue is not stimulated, meaning the glucose remains in the bloodstream. Additionally, low insulin increases glucagon release, which encourages the liver to undergo glycogenolysis and gluconeogenesis, meaning the liver perceives a fasted state and will begin converting its glycogen stores into glucose and using other non-carbohydrate substrates for energy production, further increasing blood glucose levels. Glucose metabolism also contributes to the production of oxaloacetate, which we know is an important factor in the citric acid cycle and a component of ketone body usage by muscle tissue. If there's no glucose metabolism, ketone body concentration increases and creates a state of metabolic acidosis. In the case of this young man, his initial blood samples showed a blood insulin level of 0.8, which is far below the normal control level of 4 to 8 in a fasted state, indicating a lowered ability to metabolize glucose. 1 hour post-glucose loading, the patient's insulin levels still remain low, with his level being 5 as compared to the control with a normal level between 29 and 51. This patient is also experiencing frequent urination or polyuria. This results from something referred to as osmotic diuresis. To understand what's happening and what osmotic diuresis is, it is pertinent to understand how the kidneys make urine. Urine is created when blood flows past the Bowman's capsules and the nephrons of the kidneys, and small molecules, waste, and excess water are filtered out. Unfortunately, the Bowman's capsules are nonspecific for what they filter out of passing blood. This means an amount of electrolytes and amino acids end up passing through into the forming urine. The renal tubules work to reabsorb these unintended passengers. Glucose levels in plasma create a gradient between pre-urine in the renal tubules and the plasma itself. Specific glucose-sodium membrane transport proteins are responsible for carrying glucose out of the renal tubules and back into the blood plasma. When blood plasma glucose is too high, the gradient will not allow glucose molecules to be transported back out of the renal tubules and into the blood, it will therefore remain in the renal tubules and concentrating the urine with glucose. The high concentration of glucose in the urine acts as a solute that then pulls water into the tubules via osmosis, which then is eliminated through heavy, frequent urination. Expectedly, this polyuria and osmotic diuresis leads to a net loss of systemic water. This loss of systemic water leads to an increase in blood osmolarity or solute concentration, which makes the blood more concentrated. This triggers a feeling of thirst, which then encourages us to increase our water intake, which is referred to as polydipsia, which is another of this young man's symptoms. The patient's mother also described her son as often appearing drowsy, which may be a reference to the fatigue caused by the transition of energy substrate from fat to carbohydrates. Energy metabolism is fine-tuned to use transition of energy substrate from carbohydrates to fat. Energy metabolism is fine-tuned to use carbohydrates as the most efficient and high-yield substrate. But when insulin is in low supply or non-existent, energy production transitions to fat as its source. ADP rephosphorylation is decreased and the body cannot resynthesize ATP as quickly, leading to fatigue. Based on this boy's presenting symptoms, along with his lab results, it would be safe to say this boy is most likely suffering from type 1 diabetes. Due to this patient's diagnosis of type 1 diabetes, one would expect his fasted blood glucose levels to remain elevated over the control. Because lack of insulin production is a characteristic of type 1 diabetes, it would be expected that blood glucose levels will always be elevated over a control since there is little or no insulin present to encourage glucose metabolism. In the above discussion on energy metabolism and glucose homeostasis, we began looking at some of the important regulatory hormones involved, specifically glucagon and insulin. Individuals with uncontrolled or undiagnosed type 1 diabetes cannot produce enough or any insulin. Since insulin acts as a glucagon suppressor, when it is not present, glucagon secretion proceeds unchecked. Glucagon is responsible for triggering the process of gluconeogenesis, which is the body's way of synthesizing glucose from non-carbohydrate sources. Two regulatory enzymes responsible for gluconeogenesis are phosphoenolpyruvate carboxykinase, or PEPCK, and glucose 6-phosphatase. Additionally, I looked at case number 4. In this case, when the body is in a fasted state and available blood glucose drops, it relies on the breakdown of glucose-containing compounds to maintain glucose homeostasis. One of the primary glucose storage compounds is glycogen, and it is primarily stored in the liver. When we eat, increased glucose levels in the blood stimulate the release of insulin. Additionally, high levels of glucose in the blood stimulates the liver to take in more glucose via the GLUT2 transporter in the mitochondrial membrane of the liver cells than necessary for energy production, and the enzyme hexokinase begins converting the excess glucose into glucose 6-phosphate. Insulin then stimulates glycogen synthetase to convert the glucose 6-phosphate into glycogen, while at the same time inhibiting glycogen phosphorylase enzymes. When insulin levels drop and glucagon secretion by the pancreas exceeds insulin secretion, the reverse occurs. Glycogen synthetase is inhibited and glycogen phosphorylase is activated to convert glycogen to glucose 6-phosphate, which is further hydrolyzed by glucose 6-phosphatase into glucose to be released into the bloodstream. In the presence of added epinephrine, glycogenolysis is augmented and further glycogen stores are converted to glucose for use by the brain and red blood cells. Hereditary fructose intolerance is a genetic condition characterized by the inability to break down fructose due to a deficiency of aldolase B enzyme. Patients with this disorder typically begin experiencing symptoms as they wean off breast milk and onto foods containing fructose, sucrose, and sorbitol. Symptoms of a hereditary fructose intolerance are similar to those experienced by the infant in this case study and include hypoglycemia, lactic acidosis, and hepatomegaly, or an enlarged liver. However, additional symptoms include nausea and vomiting, abdominal pain, and abdominal distension, and were not present in this infant. Additionally, as stated above, hereditary fructose intolerance occurs as an infant is weaning off breast milk and is exposed to fructose, sucrose, and sorbitol. In the case of this patient, the mother was explicit in stating this infant was only being fed breast milk and therefore had not yet been exposed to any symptom-forming compounds. This means hereditary fructose intolerance is not the correct answer. Galactosemia is another genetic disorder resulting from an enzyme deficiency. In the most common type of galactosemia, the individual lacks the enzyme galactose 1-phosphate uridyl transferase. Deficiency of this enzyme means the individual cannot break down the simple sugar galactose, a component of lactose. Patients with this disorder cannot tolerate milk of any type, be it human or animal, and symptoms of this disorder are apparent immediately. These symptoms include feeding difficulties, vomiting, poor weight gain, and in more serious cases, coagulopathies that cause bleeding, increased susceptibility to infections, jaundice, and eventual liver failure. In the case of this infant we are studying, one of the above symptoms of galactosemia in the case of the infant we are studying, none of the above symptoms of galactosemia are present aside from liver dysfunction, which does not occur unless the disorder is left untreated. We also know the patient did not begin experiencing symptoms until 2 months of age. In patients with galactosemia, symptoms are immediate upon exposure to milk. Again, galactosemia is not the appropriate diagnosis for this infant. Individuals with glucose storage disorder type 1 typically present with symptoms in early infancy, which is the case with this patient. Symptoms include those of hypoglycemia, such as jitteriness and potentially seizures. Hypoglycemia occurs because, despite being able to convert glucose to glycogen, individuals with GSD either lack the glucose 6-phosphatase enzyme needed for conversion of glucose 6-phosphate back into glucose, or they lack the transporter enzyme glucose 6-phosphate translocase. This infant began showing symptoms of hypoglycemia when it started sleeping longer between meals. This meant it was in a fasted state for longer periods of time. Either type results in an inability to fully convert glycogen to glucose to be used in the fasted state. Patients with glucose storage disease type 1 may also present with an enlarged liver due to glycogen and fat store buildups. This infant presented with an enlarged liver. When patients with glycogen storage disease 1 undergo glucagon or epinephrine challenge tests, results will show no increase in blood glucose, but instead will show an increase in serum lactic acid. This infant's blood test after an epi challenge showed just that. Based on the above, it is safe to say this patient is most likely suffering from glycogen storage disease, specifically glycogen storage disease type 1, or von Gierk's disease. Glycogen storage disease is the appropriate diagnosis. Lastly, glucose 6-phosphate dehydrogenase deficiency is another genetic condition diagnosed in infancy where a genetic miscoding leads to a lack of G6PD enzymes in the individual. Unlike the rest of the conditions above, which primarily affect digestion, this disorder affects red blood cells. G6PD is an enzyme that exists in the cytoplasm of cells and works to protect the cell from damage by reactive oxygen species. Because a vital role of red blood cells is oxygen transport, they are especially vulnerable to reactive oxygen species damage, and a deficiency in G6PD enzymes can cause acute hemolytic anemia. Episodes of hemolytic anemia occur most often when an individual with G6PD deficiency experiences an increase in reactive oxygen species, which is usually brought on by exposure to certain foods and medications. In infants, symptoms of this disorder are characterized by decreased muscle tone, sleepiness, lethargy, and jaundice within the first 24 hours after birth due to hyperbilirubinemia. In this case study, the infant is not experiencing any of these symptoms, neither has it been exposed to any medications or foods that would trigger hemolytic anemia, so this would not be the correct diagnosis.